The first person we're going to hear from this morning is Laila Weatherly from OHSU. She's a second-year medical student, and she's been assisting with the natural history study at OHSU, and she's going to give us an update on that. Thank you, Laila.
Okay. Hello, everyone. As was already said, I'm Laila Weatherly. I'm ending my second year at OHSU Medical School, and I've had the pleasure of starting to work a little bit with the Hayflick Lab, who I'm sure most of you know over there, on a kind of small research project involving people with BPAN that I'm going to be talking a little bit about today. So I've been, along with the Hayflick Lab, looking at characterizing behavior in people with BPAN, specifically repetitive behaviors. So just a little bit about kind of what we're studying and why. We've known for a long time that people with BPAN share some sort of behavioral features to those with autism spectrum disorder and might even kind of carry a dual diagnosis. And specifically, they both display what we call repetitive behaviors. And I'll talk a little bit more about kind of the specific definition of that in a second. But nobody has really formally studied repetitive behavior or behavior at all in BPAN specifically, and so we decided to take a look at behaviors in kind of a large range of people, and see if we can identify patterns of behavior in people with BPAN, especially progression over time. And so how we're doing this, I'm sure a lot of you have heard of the BPAN Ready study or are already a part of the BPAN Ready study, but every year as part of that study, the repetitive behavior scale, sorry, things are moving on their own here. There we go. You got a little glimpse, a little preview. But that survey is administered to people every year who are enrolled in the BPAN Ready study. And the repetitive behavior scale measures the presence and severity of certain problematic behaviors over time. And we're using that repetitive behavior scale to measure and track behavior in this case. So we've included 119 participants who are in the BPAN Ready study who've been filling out that repetitive behavior scale yearly for as many as 5 years, and a lot to most of people. And the reason we're doing this is to really help understand what behavioral features with BPAN, people with BPAN have and how those behaviors progress over time. And the purpose of this is really to inform families and caregivers and medical providers about what they might be able to sort of expect behaviorally as a person with BPAN ages. So here's an example of what the RBSR survey looks like, and this might be familiar to a lot of you, but it basically asks questions about specific behaviors and how often they occur and how severe they are. So whole body rocking, head rolling, flapping hands or fingers, and then the caregiver answers on a scale of 0 to 3 if that behavior occurs and if it, how severe of a problem it is. So 0 would be the behavior does not occur, all the way up until 3, meaning the behavior occurs and is a pretty severe problem. And so, higher scores indicate more severe behaviors in general. So you've probably seen this slide a couple times, but this is kind of initially some of the general patterns that we found. So we found that stereotypic behaviors, and so what that means are repetitive motions without any sort of obvious purpose, like hand flapping, body rocking, maybe repeating sounds or words. Those are really prominent in people with BPAN all across the board, which might sound a little bit familiar to you. And then similarly, restricted interests. So that would be like a limited range of focus, interest, or activity, such as the fascination with a particular subject like trains or dinosaurs. Those were also reported really frequently across the board in our sample here. Interestingly, I thought it would be fun to include the question, does your child like the same CD, tape, record, or piece of music played continually, or do they like the same movie or part of a movie or video played over and over again? Had the most yes answers of all the 43 questions that were asked in the survey. So if that sounds familiar to you, you are not alone. Right. And then kind of on the other side of the coin, compulsive behaviors we found might be a little bit less severe kind of in people with BPAN in general. So examples of that might be the need for things to be even or symmetrical, repetitive counting, or hoarding or hiding specific items. All right. And most of all, as far as kind of looking at the behavioral picture in people with BPAN, we really found that behaviors vary and range super widely, which makes sense. There's no one look or behavior profile for someone with BPAN, so it makes sense that everyone is really unique as far as that goes. When we started looking at progression of behavior over time, we did interestingly find out that a lot of people with BPAN seem to kind of grow out of a lot of these problematic behaviors over time, even if it's kind of mildly. So specifically, this means that a lot of behaviors are becoming less intrusive or disruptive to individuals and families over time. So a lot of the behaviors we looked at started to become less problematic after the age of 5. So that includes those repetitive motions like the hand flapping, nodding head, kind of repetitive throwing of objects. Those we did see in our sample to become a little bit less problematic starting at age 5. And then similarly, those restricted interests, so the fascination with one subject, liking that same song or video played over and over, were also likely to decrease, at least mildly, after age 5. And we actually expect the sort of severity scores for both of those types of behaviors to decrease by about 50% over the ages of 5 to 18. So over those 13 years, we saw those kind of cut in half based on our modeling. On the other hand, self-injurious behaviors, so like biting, maybe banging a head against a wall or a table or something, we did see those starting to become maybe a little bit more severe after age 5. However, we did see the severity of those behaviors leveling out after age 18, and a lot of people starting to get better after that time, so kind of getting worse before it starts to get a little bit better into adulthood. And then finally, resistance to change and compulsive behaviors, if they're present, those were more likely to persist into adulthood without really getting significantly better or worse over time. So that means things like refusing to visit new places, difficulty changing routine, the need for things to be even or symmetrical. Those might not necessarily decrease over time meaningfully, but based on our sample, weren't necessarily likely to become more problematic over time either. This might be a little bit hard to interpret, but I wanted to include a quick graph to help you all kind of visualize what I'm talking about. So here you can see each dot represents one of the repetitive behavior surveys that probably one of you filled out at some point in time. And then as a reminder, on the y-axis, the higher scores mean more problematic or more severe behaviors. And then each line, in theory, is connecting two dots and represents a person and their kind of slope or change in behavior over time. And so it's really important to remember that not all people are having, a decrease in the severity of behaviors over time. We do see some people increasing, there are some lines with that positive slope, but overall, when you look at the graph and once we did our statistical analysis, we could see that, the general trend was that people were becoming less severe over time, especially starting after that age of 5. And so notably, if your child or loved one is really struggling with repetitive behaviors, based on what we've seen in other people with BPAN, many of those are likely to significantly improve over time, at least mildly, based on the patterns that we've observed. With the exception of self-injury, which I talked about, might get a little bit worse kind of in adolescence and childhood, and then kind of stabilize over time. Wanted to give kind of a quick summary, just to go over all that again. So in general, we found that people with BPAN tend to struggle with those repetitive motions and restricted interest type behaviors the most, but especially those two do seem to become a little bit less severe after age 5. And I did want to include that we don't yet quite know why this is. We've talked about it a lot in the lab, and we've hypothesized that it certainly could be directly related to kind of growing older and maturing. But it's also important to note that as people with BPAN age, we also see kind of onset of different movement disorders and intensified maybe changes in cognition, which could definitely impact the ability to perform those repetitive behaviors in the first place. And so we want to keep that in mind when we're thinking about interpreting these findings. And then lastly, I just wanted to include an acknowledgement slide, mostly to thank everyone that has been involved in the BPAN Ready study and is allowing us to look at data like this and so much more. So keep filling out those surveys when you get the links every year. And then big thanks to everyone at the Hayflick Lab at OHSU. And then for those who are not already enrolled in the BPAN Ready Natural History Study, I included a QR code on the acknowledgement slide, and you're welcome to take a picture or show that to your phone camera, and it includes a link that will take you directly to the enrollment page if you're not already a part of that. This is just a really small fraction of the data that we get from that BPAN Ready study and allows us to kind of keep looking through that data and finding more interesting things. So thank you.
Up next, we're going to have Dr. Arcangela Iuso. Dr. Iuso is a senior scientist at the Institute of Neurogenomics Helmholtz Munich and the Institute of Human Genetics Technical University of Munich in Munich, Germany, and she is going to give us an update on her research. Thank you.
Good morning, everyone. Thanks for having me here today. During my talk, I'm going to give you a research update on a project I've been working over the last year and a half. Okay, so the project I've been working on was focused on investigating a particular aspect of BPAN pathogenesis, so the mitochondrial dysfunction. Everyone knows that in BPAN there is a defect of autophagy, but somehow this was a different angle we wanted to investigate. But before entering into the details, I just want to remind you what are mitochondria, why they are so important. So mitochondria are cell organelles, and most of the time they are referred to as the powerhouse of the cell, because their function is primarily that of producing energy for the cell. And this energy is produced by the activity of 5 different complexes, which are named complex 1 to 5, and the process takes the name of oxidative phosphorylation. So basically, complex 1 and 2 oxidize NADH and FADH2, and at the same time, the electrons which are recovered by this oxidation are transferred to the electron transfer chain to the oxygen, molecular oxygen, which is reduced to water. At the same time, complex 1, 3, and 4 pump protons across the mitochondrial inner membrane and form a so-called electrochemical gradient. Now, you see the last complex 5, uses the re-entry of protons through to the complex 5 into the mitochondria to drive the conversion of ADP and phosphate into ATP. And ATP is the energy molecule that is used by the cells. Why I'm telling you this? Why they are so relevant for BPAN, these mitochondria? In 2021, we developed a mouse model for BPAN. So basically, we deleted the healthy copy of the gene WDR45. And what we observed, besides obviously having signs of neurodegeneration in the brain, besides having movement disorders, visual impairment, hearing impairment, and also autistic-like behavior in this mouse, what was new in this mouse was the presence of the mitochondrial dysfunction in the brain. In particular, the complex which was affected by the defect was complex 1, so the main entry point for the OXPHOS. Instead, the heart, which is usually not affected in BPAN, was also not affected by the mitochondrial defect in our mouse model. But somehow we ask ourselves, what's the relevance for the BPAN patient? I mean, this is a mouse model, but at that time, no patient with mitochondrial dysfunction had been identified. We had to wait till last year when we find a first BPAN patient with clear mitochondrial dysfunction. But I want to tell you the story. This patient was not diagnosed initially as a BPAN patient. It was diagnosed clinically as a mitochondrial patient. The MRI of this patient did not show any iron accumulation initially. They saw the typical presentation of a patient with Lee syndrome. And also the clinical phenotype was slightly different from that of the BPAN patient. But because of the clinical presentation resembling a primary mitochondrial disorder, obviously we checked mitochondrial function in tissue which were available from the patient. So the skin cells, the fibroblasts, and skeletal muscle biopsy. And what we saw is that we found a complex 1 defect in both these tissues. And in addition to complex 1 defect, the skeletal muscle had also a complex 2 defect. But then we were surprised because when we received the data from the genetic investigation, we couldn't find any mutation in known genes associated with mitochondrial disorder. Instead, what we saw is a mutation which is associated to BPAN. It's an already known pathogenic mutation in VDR45. So this was the clear case, the first case of a BPAN patient with a mitochondrial dysfunction. But this opened a lot of questions. So the first question is mitochondrial dysfunction a typical feature of BPAN or was peculiar of this individual? The second question we asked, if there is a mitochondrial defect, is it possible maybe to modulate mitochondrial function with small compounds, at least to ameliorate this defect and contribute to ameliorate part of the pathogenesis? And then the third, but not least important question, is the brain affected? Because at that moment, we had investigated just skin cells and muscle cells, but not the brain. So to answer the first question, if mitochondrial dysfunction was a general feature of BPAN, we took fibrils from additional 2 individuals. These 2 individuals had a typical BPAN classical presentation. In particular, one had also a known pathogenic variant in VDR45. The other patient instead had a newly identified variant. And we compared then the mitochondrial function of these 2 patients with that patient already reported with complex 1 defect. And we compared it also to fibrils from healthy individuals unrelated to BPAN. What we used was an assay that is called high-resolution respirometry, but in the lab we call it Sears assay by the name of the instrument. So basically, one needs to seed fibrils in a special plate, let the cells grow until they cover the whole plate, and then this plate is inserted in the instrument, and with the instrument we can inject specific molecule to see how the respiration changes in response to these compounds. And on the right side of the slide, you can see that there is a typical trace of the Sears assay. So first we have a basal respiration, so how the mitochondria produce energy under a resting condition. And then what we did, we permeabilized the cells using digitonin. So basically, we allowed specific compounds to enter the cells. And these compounds feed complex 1 or complex 2 of the respiratory chain. And then we could see the contribution of these 2 compounds to the basal respiration. And this is in the end what we get when we analyze those data. So as you can see in the first graph, we have the basal respiration. In gray is the respiration of the control cells, and in color the respiration in the 3 patient cell lines. You can appreciate that there is reduced respiration, reduced mitochondrial function in all 3 cell lines. And when we checked for the specific contribution to the respiration by complex 1 and complex 2, we saw that there is defective complex 1 dependent respiration. This is what is labeled like glutamate malate dependent respiration. And complex 2, which is succinate dependent respiration, was affected only in one of the 3 patients. So this told us that regardless whether it's complex 1 or complex 2 or both complexes affected, there is mitochondrial dysfunction in BPAN, and this seems to be quite a general feature. Obviously, this was a trial study, a pilot study, so the number of cell lines was limited, but over 3 cell lines were all affected by mitochondrial defect. And then there was the second question. If there is mitochondrial defect, can we modulate this mitochondrial function so that we rescue at least this defect? I mean, there is multiple ways to modulate mitochondrial function, but we didn't want to use test random treatment. We wanted to have a more focused approach. So what we did is we performed some transcriptomic analysis to check which pathway was particularly downregulated or upregulated in BPAN cells. As you can see, in green and in red, there are all the genes that are differentially expressed in the patient cohort compared to the control cohort. And there are roughly 100 different genes. But to understand which pathway is really affected, then you have to feed this list of genes into a database. In our case, we use the KEGG database. And then the database tells which pathway is upregulated, so works more or works less in BPAN. And then we looked for those pathways that were correlating with mitochondrial function. You see in red, the pathway which was downregulated and was connecting it to the mitochondrial function was the cyclic AMP. Cyclic AMP is a small molecule, always present in our cells. It has multiple functions. And some of the functions are, for instance, to activate complex 1 activity through phosphorylation of specific protein belonging to the complex 1. But sometimes cyclic AMP can also increase the biogenesis of mitochondria. So you have the defect, but if you have more mitochondria, this increased number of mitochondria could compensate for the defect. Or, for instance, cyclic AMP can also remove reactive oxygen species from the cells, which are detrimental for the mitochondrial function. So we decided to use few compounds activating specifically this pathway. And we choose 3 based on our previous experience in the lab and also based on the literature. So we use B2 cyclic AMP. So basically, B2 cyclic AMP is an analog of cyclic AMP, but it's cell-permeable. And then we use also forskolin and resveratrol. These are natural compounds that are stimulating the cells to produce more cyclic AMP, or they somehow reduce the degradation of the endogenous cyclic AMP. So we treated the cells for 6 hours, and then we subjected the cells after treatment to the Sears assay. Obviously, we identified the optimum concentration for each of the 3 compounds using toxicity assay. So now you will see 3 different graphs. Each graph refers to individual cell line. So you see that the untreated cell lines has a defect in the basal respiration, and after the treatment, there is a rescue in the basal respiration with all 3 compounds. And we show the strongest, let's say, effect using forskolin and B2C AMP. And the rescue was significant with these 2 compounds in 2 patients. The last patient, the pink one, the patient with the strongest mitochondrial defect, as you can see, benefit from the treatment with these 3 compounds, but the rescue was not significant. So this means that if you have a stronger defect, you probably need to fine-tune better the duration of the treatment in order to get a better rescue. But then we went on, so we wanted to check whether these molecules exerted their rescue effect by activating complex 1. This is what we were expecting. But looking at this data, it looked like there was no, let's say, the rescue we saw was not mediated by complex 1 defect. Probably some other mechanism that takes part in these cells. As I said, can be elimination of rescue, it can be biogenesis, mitochondrial biogenesis. So we still don't know what mechanism really triggers this rescue, but there is a certain rescue. And then finally, we wanted to answer the last question. Is the brain affected? Obviously, we can't assess the brain cells from the patient, so we have to play around. So we take usually skin cells from the patient, we reprogram them into induced pluripotent stem cells, and these cells are cells that are potentially able to become any cell type of our body. And then we, using specific media and supplements, we can differentiate them into brain cells. Now, there are multiple types of brain cells, so we had to focus on 2 types. So we focused on dopaminergic neurons and astrocytes. We choose dopaminergic neurons because these are really the cells which transfer the nervous signals through the release of dopamine. And these are also the cells that are actually dying in many neurodegenerative disorders, including BPAN. And then we choose the astrocyte. These are not neuronal cells. These are so-called glial cells, and are cells that help, let's say, that feed dopaminergic neurons and help the homeostasis and the well-being of the neurons. So this is just a picture, just to give you the idea how this process works. So to go from fibrous to induced pluripotent stem cells, we need almost 1 month. And at the end of this procedure, we have to test that the quality of the iPS is really good, that these cells are really able to become any cell of the body. And for this reason, we have to check the expression of pluripotency markers. I'm showing here 4 of them, OCT4, SOX2, LIN28, and NanoG. And our iPS expressed all these biomarkers, so they were good, and we decided to move on with the project, and we differentiated into astrocyte. Again, also this process takes more than 1 month, and again, we have to validate in the end of the process that what we get are really astrocyte. We use the GFAP, which is the standard marker of the astrocyte, and our astrocyte nicely expressed this marker. So we moved on using this astrocyte for our functional assay. This time, we didn't use the same protocol we had used for the fibroblast. We used a slightly different protocol because at this moment, we knew already the contribution of complex 1 or complex 2. We just wanted to see how mitochondrial function works under a steady state condition or when we stress the mitochondria. And FCCP is the molecule that stresses mitochondria. So at the end of the measurement, we got the basal respiration and the maximal respiration. As you can appreciate, there is defective basal and maximal respiration in the astrocyte. So the astrocytes are also affected by the mitochondrial dysfunction. And then we moved on to the dopaminergic neurons. Also here is 30-40 days protocol, and we have to validate that in the end we have really dopaminergic neurons with some markers, staining with some marker, FOXA2, MAP2 to G1, and we had very good dopaminergic neurons. But here there is this differentiation is a bit tricky. So first of all, after 40 days we have young dopaminergic neuron, but if you want, you can let them age a bit longer in the dish. But for sake of time, because of the short duration of the project, we actually investigated the dopaminergic neuron when they were young. And then we realized also that in the protocol that we were using, there was the utilization of cyclic AMP. And the concentration that was supposed to be used in this protocol was twice the amount of the cyclic AMP that we had used for treating the fibroblast. And the treatment and the supplementation of cyclic AMP was for 14 days here. So we reasoned that probably using cyclic AMP in the media would have somehow masked the mitochondrial dysfunction. So we decided to proceed in parallel, differentiating cells in present and in absence of cyclic AMP. And then we measured mitochondrial respiration. This is again the basal and the maximal respiration. The first set of data is without cyclic AMP, the second set is with cyclic AMP. I guess you can see that there is no defect. And somehow this was unexpected. It was also counterintuitive. We were expecting the strongest effect in the dopaminergic neurons. Instead, we end up finding that the neurons were healthy. So we don't have the answer why this happens, but obviously we have some hypotheses. One of the hypotheses is, okay, we've been using young dopaminergic neurons. Probably if we would repeat the analysis when they're getting a bit older, probably the defect starts to appear. That was one idea. The second idea is that maybe in the neurons there are some compensatory mechanism ongoing that somehow overcome the mitochondrial defect. And this, we want to, let's say, prove this hypothesis by doing some transcriptomic or proteomics. But then there is a third hypothesis, and the hypothesis is that probably mitochondria are so essential for the differentiation of iPS into dopaminergic neurons that if you have a defect, you will never get defective dopaminergic neurons. So we basically are selecting only the healthy cells. So those cells which have healthy mitochondria, and therefore we will never find defective mitochondria in the dopaminergic neurons. Yeah, so now there are the future directions. So as I said, obviously, we want to do further validation in this direction, trying to understand why dopaminergic neurons are healthy. But on the other side, since we know that astrocytes are affected by the mitochondrial dysfunction, we want to try rescue this cell type using stimulating the cyclic AMP pathway. So with this, I conclude my presentation. I want to thank you for the attention. I want to thank you the funding, particularly NBIA, the sort of association who funded this specific project, and all the people in my lab. Thank you.
Thank you. Up next, we have Dr. Joseph Vithayathil from the Children's Hospital of Philadelphia. He works in the BPAN clinic, so many of you have probably met him before, and he's going to give us an update on some of the research going on at CHOP.
Morning, everyone, and thanks for giving us this opportunity to talk about what we've been doing at the Children's Hospital of Philadelphia. So Sherry said I'm Joe Vithayathil. I'm one of the clinicians in the BPAN clinic, and I work alongside Dr. Adang, who's been, it doesn't show up there, all right, who's also been conducting the natural history study for our BPAN patients. And so I'm here in her place kind of presenting some of the data that we've accumulated on our natural history study, and our goal is to develop the metrics that we want to use for when we're ready for clinical trials. So there's no disclosures. Our fundings are from the Don't Forget Morgan Foundation, as well as leveraging some of the funding from our Global Leukodystrophy Initiative Clinical Trial Network at CHOP as well. And a lot of the data that you're seeing is what's been generated from the work that's been done by our leukodystrophy clinical team in concert with Dr. Adang and Dr. Thakur also when she was at CHOP just a few years ago. All right, so quickly just to kind of highlight the teams that have been involved in this. As I was mentioned, this is our Leukodystrophy Center of Excellence, and we've been leveraging a lot of the resources that they've had in their clinical trial studies and their network and the folks that help in that clinic to help us with our BPAN clinic. And then on the right side is all the members of our BPAN clinic, and we were made an NBI Center of Excellence last year, and so that was sort of the ceremony there, and it was nice to have everybody involved. And so as far as the clinic, we launched the clinic in 2021, and as I mentioned, we've been now an NBI Center of Excellence. And so over the last couple of years where I've been in the clinic, we've amassed quite a number of BPAN patients, and so the total number of patients that we've been seeing in clinic since the inception of the clinic is, these are kind of rough numbers, but around 75 to 80, and actually that might be underestimating some of them. And then last 2 years since I've been there, we've been getting about 20 to 22 new patients a year, which has been great, and that's we very much appreciate all the participation of the patients here that come out to Philadelphia and come to our clinic. And then we're recently going to be developing collaboration at Houston with Dr. Thakur setting up her BPAN clinic there. So now to get into some of the clinical research that we've completed. The main goal for our natural history study is really to try to optimize our readiness for clinical trials. And so as far as the development of therapies for this patient population, there's obviously the therapy development part, which is some of the basic science work that you've heard about in looking at how can we target specific cell types, specific organelles, and how to specifically target the dysfunction, either with small molecules, gene therapy, et cetera. But the other question we've got to be ready, and the other issue that we've got to tackle at the same time and be ready for is what to look for when we run these clinical trials. And that kind of translates to what's the patient population that's going to be best suited to test these therapies what are the outcomes that we want to measure to be able to tell that these treatments are successful, and overall in concert is making sure that these therapies are going to be applicable to the community as a whole and not necessarily just a specific population or subset of the population. And so the general approach for our natural history study has been doing both retrospective and prospective data collection. And so retrospective is basically going back in time to look at symptom development and progression, and then prospective is really looking at the time that we see the patient and from the diagnosis and how the symptoms progress from that time point. And so with most prospective studies, what you're getting is at the time of diagnosis or at the presentation to that specific, when they enroll in that study, you're collecting all the data from that point onward. So you're missing a big chunk of the data of when the patients were developing symptoms that weren't being documented up until the point of diagnosis. And with retrospective data, you're obviously you're missing any of the progressive information that's coming from the diagnosis. So by combining both of these approaches, we're hoping to get a nice longitudinal analysis of what happens from the onset of symptoms to later in life. And so, and part of this is really filling this, again, like I mentioned, this diagnostic gap of where the symptom starts, where the diagnosis happens, and on this chart on the left, you can see with many patients, there's a variability from when they present with symptoms to when the initial diagnosis gets made. And so with our retrospective data collection, we're able to capture a lot of that information. And so why is this important? So the role of natural history studies is very critical for some of these clinical trials for rare disorders. Because when you think about doing clinical trials, it's going to be very hard to do randomized control studies, which are sort of the gold standard for how we do treatments in clinical trials in the clinical research field. But with rare disorders, you're not going to have enough numbers of patients to be able to do that type of trial. And so the natural history studies are important because they actually can serve as control populations for when we do when we perform clinical trials. And so by having these natural history studies, we're able to see what the longitudinal progression and history of the disorder is, and in a clinical trial, then using a subset of patients, we're able to compare it to the data that we have from the natural history study. And so what we've been doing in the clinic is basically when patients come in, we've been doing sample collection both of serum as well as, and now collecting urine for eventually looking for biomarkers, but currently it's mostly doing delineating some of the outcome measures. And so we have neuropsych testing as well as PT evaluations and occupational therapy evaluations. And then we also collect a lot of the charts and retrospective data from prior clinical visits to be able to outline different neurologic milestones and symptoms and their progression. And so that happens through a standard kind of extraction of standard process for extracting the data from the clinical records, and then doing rigorous statistical analysis on the results to be able to come up with metrics that seem like they will be helpful for clinical trials. And so some of the initial studies, the work that came out of this natural history study from Dr. Adang was a lot of the retrospective data where we were characterizing the progression and the retrospective symptom progression in patients, and kind of look at differences between males and females and quantify some of these changes very specifically. And so these graphs on the left kind of highlight essentially look at what percentage of patients meet these different developmental milestones, with the one on the left being, with the blue bars and horizontal bars being females, and the one on the right being males. And so given WDR45 is X-linked, we do see sort of this pattern where the females tend to have better motor development skills, which are where you see a lot of the red and pink bars that are on that extreme left graph. And then with speech development, there tends to be more of a significant delay that's there. And then we do see that with the male cohort of patients that some of those development milestones, particularly with the motor development, are also continue to be significantly delayed. And this was, we kind of represented this in a couple different ways, and so the graphs on the right kind of represent them in a graph that's called like the Kaplan-Meier chart. And so the females are in the blue lines that tend to see more development or some more motor development. The male lines tend to plateau a little bit earlier. And then one of the other things that we had identified was also there seemed to be a slightly difference in separation in the patient population if you looked at when they first presented with their symptoms, with patients that had very early developmental delays that were before 6 months of age, that they tended to have a little bit slower motor development progression, and a little bit more severe language development. And so their language development tended to plateau a little bit earlier, and their motor development tend to be a little bit delayed. And so the motor development is on the graph on A, where you can see that the blue bar is the patients, or the darker bar is the patients with earlier onset. And even though they do eventually progress, it is a little bit more delayed compared to the patients that have a little bit later onset of symptoms. Whereas for the language, you can see that with the early onset, there tends to be a little bit earlier plateau of language development compared to the patients with later onset symptoms. Interestingly, the development of seizures in both these populations seems to be equivalent. And so the main goal now has really been now trying to get some of the prospective measures and actually get some of these outcome measures that are going to be very relevant for clinical trial studies. And so what we've been doing is really applying a variety of different validated outcome measures, and we've been using tools that have been used in the cerebral palsy field or in the field of autism spectrum disorders to measure some of these metrics. And so one of the ones we're using is called the GMFM66, which is the gross motor functional measure. And this is a 66-item sort of question list for those that haven't been to the clinic. And it helps get a range of motor development skills in patients and see what their developmental progress in that domain has been. And so this is in a subset of the patients, although we've now collected a lot more data on patients that we're putting together, more information that we'll hopefully be able to publish on and add to this data set. But what was nice with the GMFM66 is that we get a nice ability to be able to quantify some of the motor development in a lot of our patients with BPAN. The P-body 2 is a metric to look for fine motor skills. And so this was also a useful task where we were able to get reasonable measures of these skills in patients. And you can see in this one, the triangles represent patients with those early onset symptoms versus the later onset. And in some of those fine motor skills, there seems to be separation, although with this initial pilot data set, it's a little bit hard to draw statistical conclusions. And this, we tried to do some more of the cognitive skill assessments, and this was using something called the Leiter 3 percentile or Leiter 3 assessment, which is a way to do nonverbal IQ assessments. And what this highlights is sort of one of the things we're trying to avoid with some of the assessment and outcome measures we're looking at is even though we were able to score, get a score for a lot of the patients, you can see on the right side of that, where that dotted line in that graph is, a lot of the bars when we try to look at the percentile scoring is very, very low. And this is something called the floor effect, where basically we're not able to get a dynamic range of the data, which is problematic because then if you're doing some sort of intervention to see if it's having an effect, if all the patients are universally aren't able to do the task, then you're not going to be able to see if unless there's obviously some huge effect, you'd be able to see that there's an improvement, but some of those effects are not as typical, and so you're trying to find something where you're getting some responses where you're going to have a dynamic range of outcomes that you're going to be able to see a difference when you do an intervention. And one of the limitations with this IQ test was that many of our patients have a short attention span and focus, and so it was always, when we were in the patient interaction group, it was very clear, it was obvious with all the BPAN, the kids with BPAN, where they were immediately getting up from the tables and wanting to walk around and play with each other. And so that tends to happen in our clinic as well. And so this test was very limited by some of those attention issues, so it's not going to be a great outcome measure to use when we eventually go to conduct clinical trials. But what was more helpful was something called the Vineland Adaptive Behavioral Scale, which is another way, it's a patient survey and parent survey, and this really provided us a lot more dynamic range of responses, and we were able to assess a lot more functional abilities in our patients that weren't being picked up by that previous outcome measure. And more recently, this past year, we've published on data looking at really a quality of life outcome measures. So it's great that we have some of these outcome measures to look at functional assessments, but what's really important is also being able to tell in families like yourselves what's actually important to you from a quality of life standpoint. And so we had used a variety of different quality of life assessments. And this one is called the CP Child, which is, again, derived from the cerebral palsy literature, but is a nice overall assessment of different aspects of quality of life. And so this was very helpful to do because we were able to identify sort of the key metrics from your perspective and the family's perspectives as what's important in your day-to-day life in terms of the functioning of your child and what helps in the quality of life. And so being able to look at that, you can see that even though a lot of our metrics are looking at gross motor development, the overall rank of that of movement and independent movement is fairly low, but potential is number 7 on the scale. But there are other metrics that are important. So these are also going to be very critical for us to be able to look at to be able to tell that whatever clinical intervention we're doing is actually effective, is if it's helping the families themselves. And so overall, we've quantified some of these disease trajectory, looking at early development of symptoms versus late development, and identified some preliminary outcome measures that potentially are going to be very useful in the clinical trial setting. And in the process of doing those in a broader population base to really validate those as good metrics for clinical trials. And then with some of the ongoing therapy development, we'll be looking for biomarker development and gene therapy development as well. And so hopefully in 2 years, we'll have more data to report on that. And with that, I'll just like to thank all the families that have participated and the rest of and everybody else that's part of the BPAN clinic. Thank you. Yeah, so everybody can, we're open to enroll everybody in our natural history study, and I should have done a QR code before, but that's the disadvantage of growing up after the not being a millennial, but I will, we'll provide some numbers and contact information to be able to enroll in our clinic, and you can always come find me and let me know, and we'll get everyone enrolled in the clinic in the natural history study. Thank you.
Thank you so much. And one thing I know that Dr. Joe and Dr. Adang are very excited about is that people also have the option to enroll remotely, so they don't physically have to go to the clinic at CHOP. Next up, we are going to have a virtual Zoom presentation from Dr. Bertrand Mollereau. He is the head of the Regulated Cell Death and Genetics of Neurodegeneration Group at the Laboratory of Biology and Modeling of the Cell in Lyon, France. And I'm going to turn it over to the Zoom as soon as it's ready to go.
Okay, so hello everyone, ready to start. So thanks. First, I would like to thank the organizer for giving me the opportunity to present our work entitled the Drosophila Model of BPAN. So this work has been conducted in our laboratory at the INS of Lyon in France, and with our team, and in particular, I will show the work of 2 PhD students, Marion Sell and Sarah Nurtent, who are supervised by Ludivine Walter and myself. So as you heard previously, I mean, the BPAN is caused by is a genetic disease caused by mutation in WDR45. I'm just going to put the pointer and we are good. Okay. And as you can see here, WDR45. So it's described now, as you heard earlier, that associated with this mutation, there are some autophagy defects. I don't have to define what is autophagy. You heard a lot earlier today. It's believed that those dysfunction, at least in part, cause the phenotypes which are observed, the clinical manifestations which are observed during the disease, not only during childhood, with, for example, epilepsy and developmental delay, but also during adulthood manifestation like motor defect and early Parkinsonism. This is a view of autophagy. You heard already earlier that WDR45 is important at different steps in autophagy. So works from the last years have allowed to, getting this to work, yes, have allowed to make major advances into understanding the phenotype of WDR45 mutation, in particular models in human cells, yeast, dictyostelium, but also mice have shown that autophagy is observed in those models, at least most of them, not all, but most of them. You can also demonstrate mitochondrial dysfunction and iron overload. The mouse model has been also very important with those for those aspects, in particular, in addition, measuring neurodegeneration, locomotor defect, and social disorder. Although there are many studies now, we still have a little understanding of the cause of the disease, and in particular, if the autophagy defects are responsible for all the other manifestations. So for that, we decided to use Drosophila, which is a fly model, which actually has been used quite widely to study neurodegenerative disease in the past, such as Parkinson, and found many important mechanisms in Parkinson disease. And maybe more recently, you heard the work of Professor Sibon, Odi Sibon, and Suzanne Flick on BPAN disease and the importance of Drosophila in this model. So we decided that Drosophila could be really of some help to address the important question, but also Drosophila is also a great model to test pharmacological compounds, hoping to find better treatment, of course. So to move on with this, in our lab, we created a fly model of BPAN. And for that, we cloned, we identified a putative homologue of WDR45 in the fly. And our first goal was basically to create a knockout model of those flies. So the idea is to inactivate this gene and look if in those flies which do not carry this gene, we see similar phenotype as in human. And among the phenotype, we can look at a phenotype which are associated with early neurodevelopmental manifestation or phenotypes, or late phenotypes such as motor defects. So, I mean, or lifespan, actually, that's what we basically, I will describe you today, is that those phenotypes ranging from locomotion, lifespan, and epilepsy. We can also look at cellular level, autophagy, and also the accumulation of iron, as you will see. I mean, our goal is basically to demonstrate that the fly mutated for this gene is a relevant model to study BPAN. That's the first demonstration that we'd like to make. So first, locomotion, and we can do that quite nicely here. You put the flies in a tubes, and then you let them climb up spontaneously. The flies climb up in the vial, and you let them climb. So as you can see here, experiment done by Marion. She tapped the fly on the left side. There are the healthy flies, and on the right side, you have the BPAN flies. I mean, you can see the healthy flies, they climb quite rapidly to the top of the vial, while the BPAN flies takes much longer. We can quantify this, and you can see that at 1 day old flies, you have a decline of performance index. So basically they climb, but much slowly. And it's true at day 21, day 22. So we can conclude that this is a progressive locomotor defect. And so when you have this, and you're quite happy with this observation, and you want to show, because BPAN is a neurological disease, can you rescue this locomotor defect by re-expressing a wild-type form of this gene in the neurons? This is kind of a genic therapy that we can do to the flies by re-expressing the normal copy of CG11975 in the fly. So we can do that. And as you can see, it works quite well. You re-express with this neuronal driver, CG11975, and you can see that now they climb normally to the top of the vial. And this work has been described in our article, which is now available on bioRxiv if you want to have a look. And an important experiment is actually we can also reintroduce not the fly gene, but the human gene in the fly. So here we bring the human gene in the neuron of the fly, and you can see also that they rescue their locomotion. So this is a very important experiment, which is telling us that the human WDR45 can work in the fly and do function of CG11975. So this is a strong point to say that this is a relevant model to study WDR45 in the fly. So with that, we can move on and call this 11975, we can call it D for Drosophila WDR45 for the rest of the presentation. And so next, we wanted to assess other aspect of the phenotype, and in particular, lifespan, which has been, was predicted to happen at reduced lifespan in the fly when they carry this mutation. So we looked at the lifespan, and as you can see in red, the fly live much shorter time, like 20 days, or here 15 days at 50%. At 15 days, 50% of the fly dies compared to control. It's more about around 50 days. So again, so another phenotype which is important, and we went on with the characterization and asked whether the flies could show some epileptic-like phenotypes. Been reported that the flies can be studied. We can study epilepsy in flies with several mutants which are similar in human and flies. So we wanted to check whether those flies could show also some epileptic-like phenotype. And for that, Sarah used this protocol in which she brings strong shaking of the flies 3 times, and after she looked at the flies for an epileptic-like phenotype, and she counts the phenotype. So as you can see, after shaking the flies, they have like a kind of a crisis. They jump everywhere. They have this loss of posture, leg abnormal, very fast legs movement, and the wing buzzing. That's what basically we can observe in those flies. So we can quantify that, and we see that the flies, indeed, the flies that are BPAN mutant or WDR45 mutant, sorry, they see this phenotype similar to one of the classical epileptic-like mutant in flies, BSS1. So with that, we have like 2 important phenotype, additional phenotype that we can characterize. And now, of course, we can ask questions. So whether autophagy, for example, is important in those for those phenotypes. Whether autophagy is important for the longevity phenotype, and whether autophagy is important for the epileptic phenotype. That's what I'm going to show you. And for that, of course, we can first show that at the molecular level, the flies exhibit autophagy defect. We have this marker, which is well used also in mammalian, LC31, LC32, which is also called ATG8. So we measure this ATG8. As you can see, the flies at the cellular level exhibit autophagy defect. So what we can do is basically activate autophagy in the flies. So we can activate, reinitiate or induce more autophagy, and we can do it here, for example, at the initiation level by overexpressing one of these important factors, ATG1, for example. And that's what Marion did. She re-expressed ATG1 in the fly, and this is the fly which carried a mutation, as you can see. And when you re-express in a dotted line here, red, ATG1, you can see there is a nice improvement, increase of the longevity of the lifespan of the fly. So that was actually a good news for the project, that you can really, by inducing more autophagy, you can increase the longevity of the flies. Next, we actually had a chance to collaborate with a group of Apostolos Papandreou, who gave us some compound that he already tested in a human cell, in particular in iPS cells, and in his paper published in 2024. He had tested several drugs, several compounds which are known to activate autophagy. Actually, we tested one of them that I'm going to show you today, which is the cardiac glycoside digoxin, which have been shown to improve cellular health in these iPS cells derived from BPAN patient cells. So in the fly, as you can see, in particular here in this context, WDR45 heterozygous digoxin treatment could improve the longevity. Here is the hazard ratio. So it's a way to calculate that. So you can see that there is a less hazard ratio when you have a digoxin treatment, indicating that digoxin can provide some improvement. You can see that the homozygous mutants do not show this protection effect. So maybe possibly due to too strong impairment of autophagy in those flies. So then moving on with more recent data, which are not published yet. And Sarah, she did also interesting experiments, similar experiments, but using another tool to activate autophagy. And actually, she didn't use pharmacological. She used genetic induction overexpression of the LAMP2A protein. LAMP2A is a lysosomal protein. So presumably, LAMP2A will allow more elimination of these autophagic vacuoles here that you are here, which are stuck probably in the cells. And when she overexpressed this LAMP2A, as you can see here in the mutant, heterozygous or homozygous, you have a nice reduction of this epileptic phenotype that she was recording. So we believe that this overexpression is sufficient to boost autophagy. So I think like in this condition, it gives us also an idea that activating autophagy downstream of the pathway may be very beneficial for the flies and for the disease, of course, to improve the phenotype and the manifestation. So almost done with the presentation, and basically to recapitulate, we think we have a valuable model of BPAN disease. As you saw, that activation of autophagy could improve lifespan and epilepsy. We had experiment that also tried for the locomotion, and interestingly, autophagy activation could not restore locomotor defects, suggesting that there are other mechanisms, other pathways which are important for the locomotion, which is not autophagy. So I didn't tell you about iron, but we also have a characterization of iron in our model, and the slide shows iron accumulation. So what is next with this model? So we want to test more drugs, because more pharmacological compounds. And for that, we are collaborating with several groups. The group of Apostolos Papandreou I told you about autophagy inducers. The group of Marion Falabrègue and about the iron, and the group of Jean-Paul Lasserre for mitochondrial activating drugs. So we can test those molecules in the fly using our assays. And we also have a program collecting cells from BPAN patients, so where we can also test the drugs in our patient cells. So that's what we want to do on the drug screening part. And of course, the fly model can help us also understanding more precisely what is the role of autophagy in all those phenotypes that we described, and also other dysfunction, possibly iron, or you heard about iron, mitochondrial defect or mitochondrial metabolism, which could be also important to explain those defects. So with that, I would like to thank you and thank people who did the work, in particular, as you say, Marion and Sarah, the PhD students, supervised by Ludivine Walter and myself. I would like to thank the founders. A very big thank to the French Association for BPAN Patient, Autour du BPAN and BPAN France. Also, $1 Million Bike Ride in the US. Many thanks for everybody. It was really a great help to achieve what we have done. I would like to mention lastly that we have an ongoing application for ERD Rare Grant, a rare disease from the European Research Alliance. We have gathered a consortium, Act on BPAN, with, as we can see, several researchers, including speakers that you have heard during the presentation, as well as 5 European patient associations. So we are now in the second phase, and we hope it will be successful to help understanding this disease and found new molecules, new drugs for BPAN. With that, I'd like to thank you all. Thanks for your support, and thanks for the organizer again. And I wish you all the best and good luck for the end of this meeting. And with that, say you bye bye, and hope to see you in person another time. Thank you very much. Bye bye.
Good morning from the Netherlands. I hope you have a great time in Greenville. Sorry that I cannot be there, but I will give you now a short summary of what we are doing here in the Netherlands with regards to BPAN research. So I will first give you a rationale for the research approach that we are doing, and then I will summarize the findings with regards to autophagy, and then I will also look a little bit what we found about what lays beyond autophagy, and I will give you a short outlook of what we are doing in the future and our ongoing work. So what we know for a fact is that WD45 protein is unfunctional in BPAN patients. And WD45 plays a role in many cellular pathways, and thereby by making them unfunctional, which will lead eventually to symptoms of the BPAN disease. Now the solution of that is we can either repair the WD45 gene or protein directly, or we can fix the pathways in which WD45 plays a role in. And since we are a cell biology lab, we are mostly working on fixing the pathways where WD45 is involved in. So, and the approach that we take is actually twofold. On the one hand, we want to gather more knowledge to really identify the pathway in which WD45 plays a role in, and also characterize precisely the role, because this would then eventually help us to more specifically target exactly this role. But on the other hand, and I will come to that in the end, we'll also try to identify compounds that can actually bypass the defects in WD45 deficient cells, because this would then just circumvent all the defects of WD45. Not necessarily cure the disease, but it helps to ameliorate the symptoms. So, in my opinion, there are basically different model systems how we can study BPAN. We have the animal models, in which, for instance, for mice and Drosophila, we can study the disease in a more physiological context. And depending on when the video is shown, either you have heard a lot of that already by Bertrand or Arcangela, or you will, I think. Or you can study patient-derived cells, and this is then more studying really the patient-specific mutations and in a patient context. Or you can use a laboratory cell line to study more the function of the protein. These cell lines are much easier to handle, and and can be quite useful, I think, to study the general function of the proteins. So we are studying within these laboratory cell line systems, and in order to mimic BPAN patient situation to a certain degree, we can now deplete, as you can see here, we can deplete the WD45 protein from these cells. Can do it here and here, and this would mimic so-called a missense mutation, in which also the patients do not express any WD45 in the cells. And what we are now currently using, we have two different cell lines, but we are using mostly these SHSY5Y cells. In our laboratory, we call them sushi cells. And the interesting thing about those are these are derived from a brain tumor, from a neuroblastoma. And what we can do now with these cells, what you can see here on this video, is that we can differentiate. It's called within a time period of 6 days, we can differentiate them into neuronal cells. So we have then undifferentiated cells, and after 6 days, these cells, by treating them with a different cocktail of medium, they really form here these connections, these dendritic connections, and more represent more a neuronal like state, which is of course also the cell type that is mostly affected in a BPAN. So we can now compare the differentiated cell types versus the non-differentiated cell types, and see also whether there are differences in the response towards WD45 depletion. So, and to identify the pathways in which WD45 plays a role in, we also focused first on autophagy, because I'm coming originally from really autophagy lab, where we study a lot of autophagy. And autophagy is a pathway, is a recycling pathway, in which is centered to this pathway is the so-called autophagosome. And I always compare this autophagosome with a sort of vacuum cleaner, because here in the autophagosome, all the sort of damaged or dirty components of the cells are basically sequestered and captured, and then they are not, they cannot sort of damage the cell further once they are in this vesicle or in the vacuum cleaner. And then this vesicle can then fuse eventually with a organelle called lysosome, and in these lysosomes there are the enzymes, and these are sort of the scissors that can actually degrade or cut these components of the autophagosome into smaller pieces. And then these building blocks that are then produced in this autolysosome or in the lysosomes, they can be recycled back to the cell, and the cell can thereby make new organs, new proteins, whatever it's required in this situation. And that is of course very handy when it comes to stress situation, or when the cell experience damaging, so that basically the components in the cells that are damaged cannot really harm the cells. And we can now, when we now want to look at how the status of this autophagy pathway is in the WD45 cells, that knockout cells, we can actually measure then the autophagic activity by measuring a protein which is called LC3-2, and this LC3 sits in the membrane of the autophagosome. You can see that here, and basically when we now quantify the amount of this LC3-2, we get a proxy of the amount of autophagosomes within this cell. And this is what you can see here. You can see that the columns here are basically represent the amount of this LC3-2, which is this protein that we can see by Western blot here. And what you can already see, so this wild type on control cells are the black arrows here, the black bars, and the black bars are much higher, as you can appreciate, than the purple or blue bars, which represents the WD45 knockout cells. So meaning that already the activity of autophagy is reduced in these cells. And for us, what's very interesting is that when we now try to induce autophagy, so there are some stimuli that we can use to really trigger the pathway, and this is here called with a P because of the name of the inducer. That's not relevant so much, but you can see here in the wild type cells we get really a nice increase in this response, whereas in the knockout cells this increase, you can see here, is actually drastically reduced. So this was very intriguing to us, that sort of these so-called canonical autophagy inducers can somehow are unable to really induce the response anymore. So what we basically then found that in these cells there is a reduced autophagy flux, as we call, so the pathway is really reduced. And we see also that especially in neuronal-like cells, this defect is much more pronounced compared to other cells that are not differentiated into neuronal cells. And then the conventional autophagy inducers, they cannot initiate this autophagy response in these cells too much. And this is why we also have, with leading Bertrand as a leading scientist, together with Arcangela and others, we're trying to really find now autophagy inducers that can still sort of bypass this autophagy defect in WD45 knockout cells. And but our lab is certainly not the only one. You might have heard from Bertrand and Arcangela that they are also working on autophagy, and there are also many other labs. This list is not even full and comprehensive, but what all these researchers have found is basically that WD45 plays actually a role, I think, in two parts of the autophagy pathway. On the one hand, it's required for the fusion from this autophagosome, from this vacuum cleaner, to the lysosome. So this fusion is blocked when WD45 is blocked. But also, and it's not depicted here, it has actually also a role in making this early autophagosome structure. So at least it helps make these early autophagosome structures by pumping actually fat, so lipids, to these early autophagosome structures. So, but as I said, we are not looking at only at autophagy. We also looking what lays beyond autophagy. So basically, what is in these cells is defected that does not really belong to the autophagy pathway. And why we did that is because when we had the cells, these knockout cells, and we compared the knockout cells to the wild type cells. And this was also funded by the $1 Million Bike Ride. And what we did a so-called protein analysis, a mass spectrometry analysis, where we can now measure which proteins are more abundant in WD45 knockout cells, in green, and which proteins are less abundant, so reduced, in WD45 knockout cells. And you can imagine, we see really a few proteins that are more abundant in WD45 knockout cells, but there are also many proteins that are so-called downregulated in WD45 knockout cells. And this is very interesting to us because this would then basically reflect these proteins. If something is downregulated, at least in my opinion, it's then the idea that these proteins do not function as good. And if they are involved in a certain pathway, I would say that this pathway is also not as functional as in the wild type cells. And what we can do now is we can now take all these proteins that we consider as significantly downregulated in the knockout cells, and we can map them. And what we see, you get then this sort of so-called protein interacting map, and you see what other studies have found, how these proteins are related to each other. And when you then do a pathway analysis, we can actually see that there is a particularly sort of some proteins that really are closely working together, it seems. in that situation. And when we now zoom in and look which are or what are these proteins, we realize that these proteins are in fact all mitochondrial proteins. They play all a role in the mitochondrial biology. And knowing that, so we then looked at really the mitochondrial morphology and the mitochondrial network that it's made in the cell, and we realized that indeed this mitochondria seems to have a defect in WD45 knockout cells. And mitochondria are energy producing organelles in the cell, so they produce the energy. And how they do that is they have a system in the mitochondrial membrane, and you see a mitochondria has two membranes, an outer membrane and an inner membrane, and in this inner membrane they are sitting sort of four complexes. They are all belonging to a so-called electron transport chain, and what they do is they pump protons into this inner membrane space between the outer and the inner membrane, and then there's really a lot of they form then a proton gradient with a high proton density in this inner space, and this proton gradient then basically forces a so-called ATPase to function, and these ATPase then produces ATP, and the ATP is actually the energy of the cells, and the more ATP is produced, the more energy the cell has. And what we found is that indeed proteins of this electron transport chain are reduced in WD45 knockout cells, and especially from this complex 4, where we see a protein which is reduced, which is called COX-4, and we validated that this is reduced in Western blot analysis in our cells. And what the consequence of that is that if you have now reduced protein in this electron transport chain, you get a reduced proton gradient and a reduced energy production in these cells. And what then the consequences of these reduced energy is, what we found in the cells, we found that the growth of the cells, which requires of course a lot of energy, is reduced in the WD45 knockout cells, as you can see here. So you see here that when we grow the cells, the plata is completely full in the wild type cells, whereas it's really less full in these knockout cells. So the growth of the WD45 knockout cells is reduced. There is a defect there. And this is not the only phenotype. We also found other phenotypes, but for sake of time, I will keep it to that phenotype here. Because what we
ask if you can hear us.
Because we will also use this phenotype for the next part, which is the screening part. So now we want to identify actually compounds that can bypass these defects in these WD45 deficient cells.
Can you hear us?
And in order to do that, we need to have our analysis system as automated and as unbiased as possible. So we now established a method where we can actually automatically score or mask these cells. So you see here are the cells when they are really seated very in low density. The system now detects each cell and marks them with this yellow sort of overlay mask, and we can now follow this over time, so-called in the so-called live cell microscopy, and we can actually now follow. You see when the cells divide, there is more yellow, and we can see that also following here in these curves. And what we clearly see is as over time the wild type cells, they grow much faster than the knockout cells. And this is actually now very important, because now this phenotype in this automatic system we can now use to actually treat now these knockout cells with different compounds, with mitochondria stimulating compounds, and see whether we can reverse this growth defect. And this is what we are currently doing, and this is also funded by the $1 Million Bike Ride, and we are trying to find now compounds that can actually reverse this phenotype in order to basically found compounds to bypass the WD45 defect in the cells. Now, in parallel to that, we also want to actually now bring these, translate these findings from this laboratory cell line to more patient-derived cells. And here, together with Susan Hayflick for the collaboration and also Lena Bubula, we are doing that, and we also try to do now to use patient fibroblast cell lines and directly transform them into neuronal cells, to then validate in these neuronal cells also the findings that we have in these laboratory cell lines. So with this, I wanna finish here with our short summary of what we are doing with regards to the BPAN research in our lab. I wanna especially thank you for your attention, and also thank you for helping actually funding these projects over the years. I'm very grateful for that. Without you, it would not be possible to do any research on BPAN here. So it's very appreciated. I also want to thank all the team members over the years, very talented students who brought their effort into this work, and also the project partners that helped really bringing this research forward. And with this, I wish you all the best, and I hope I see you soon, and have very much fun here in Greenville with the rest of the conference. Bye bye.